This screencast uses the paper published by Jean Soulier and Professor Lowndes in Current Biology in 1999. It has the rather long title of the BRCT domain of the Escherichia checkpoint protein Rad9 mediates a Rad9-Rad9 interaction after DNA damage. Make sure that you have it ready so that we'll be able to discuss some of the figures in it. Okay, so let's say that we're writing a scientific paper or a thesis. First, let's think about what we're trying to achieve with the data figure. What we have is our primary data that we've collected. This is the building block of our science, and we want to take this data and try to interpret it and explain it for the people who are reading our thesis or our paper. Obviously, we need a clear and consistent way to refer to that data. That's just like the process of citing other people's papers in our thesis or project. For this reason, there's a more or less standard way to display all biochemical data in scientific publications. So let's have a look at Professor Lowndes' paper and see how it's done. Let's have a look at figures 1 and 2 on page 552. That's the second page of the article. First, notice they're clearly numbered 1 and 2. By convention, we always use Arabic numerals to number figures. We number the figures in the order which they're first referred to in the text. This is an important point. All figures must be referred to explicitly from the text. We're not simply writing for Hello Magazine, where we plonk random pictures beside our text. We actually are pointing from our text to our data and back. Figures 1 and 2 both have several parts or panels. This is for two reasons. First, most journals have a limit of the number of figures, typically between 3 and 7, so using multiple parts allows us to sneak more of our data into a paper. Secondly, and more importantly, multi-part figures allow us to group together our data. This might be done because we have variations of similar data, or, such as in the case of figure 1, we have data and we want to put a schematic overview beside it, or, as in the case of figure 3, we have the same samples and they're being analyzed in different ways. By convention, we always define the different figure parts or panels using letters. So in figure 2, you'll see that we have panels A, B, and C. Let's focus in on figure 2, panel B. This plot has all the correct layout items you'd expect for a graph except one. Notice that despite what you've always been taught, there is no title. Titles go in the legend, which we'll get to in a minute. This saves space and keeps things uncluttered. Of course, the axes are clearly numbered with labels and units like you'd expect. Notice the clear use of symbols. There are 6 overlaid curves, but it's still easy to see the patterns. For example, the -UV and +UV have separate symbol types, circles and squares. Note also the concise and succinct use of -UV and +UV to denote the undamaged control and UV damaged samples. Similarly, the nomenclature for the mutants, Rad9 delta with the triangle and Rad9F plus W for point mutants, were defined in the schematic in figure 1A. This saves space and makes the figure very clear and efficient to understand. In figure 2, panel C, we have a Western blot of an SDS polyacrylamide gel. This is also a good example of clear and efficient labeling. Across the top we have each lane labeled for its sample. Such lane labels should be at the top, never underneath. Note the use of minus and plus to show with and without UV treatment. This is a very common and efficient way of distinguishing untreated control and treated experiment samples. The different paired samples are then grouped with a short horizontal line. Above the line, the various mutant strain sources are denoted using the code defined in figure 1A. It is essential to give a size scale on every gel figure. In a normal Coomassie-stained protein gel, or an agarose gel of DNA, you will have a marker ladder. Relevant marker bands should be labeled with their size. In the Western blot in figure 2C, the sizes are determined indirectly, but they're always shown nevertheless. Note also the unit kDa for kilodaltons is shown. Numbers have no meaning without their units, so the unit must be shown. On the right-hand side of figure 2C, there are a number of explanatory labels. These indicate the unphosphorylated and phosphorylated forms of Rad9 and Rad53 proteins using simple self-explanatory codes. If you look carefully, you will see that the labels for Rad9 are slightly shifted downwards. This small error took me a couple of minutes to work out. It illustrates the importance of care and checking when preparing figures. All gel figures need labels for sample lanes and size indicators, as we've shown. Many gel figures also have simple lane numbers below the gel. Soulier and Lowndes did not use gel lane numbering like this. However, it's often a good idea because it makes features easier to refer to. For example, with lane numbers it would be possible to refer to the phosphorylated Rad9 in Figure 2, Panel C, Lane 2 very easily. The legend is the explanatory text for a figure. Every figure must have its own individual legend. This legend appears below the figure. The aim of the legend is to provide full details which complement the labeling of the figure itself and make it self-standing. It should not be necessary to refer to the main body of the paper to understand the figure with its legend. In fact, most scientists first simply read the figures when reading a manuscript. Only if they're very interested will they read the main text in detail. Many research groups run their own small seminars called journal clubs, where they sit down and discuss interesting articles. Usually the person who is presenting an article will just show the figures with the legends to lead the discussion. This shows how important it is for figures to be well designed and labelled so that they stand on their own. Let's look at the legend for Figure 2. Every legend starts with a concise one-sentence title summarizing the figure. In Figure 2 we are therefore told that the figure is about the effects of normal levels of expression of the BRCT mutated Rad9 proteins on survival, the G2M checkpoint, and hyperphosphorylation after damage. Each part of the figure is then individually described. For example, for Figure 2, Part B, we are told G2 checkpoint phenotype analysis of the Rad9 F+W mutant. Cells that arrest at the G2 checkpoint are large, budded, and with a single nucleus. Error bars have been omitted for clarity, but standard deviations from at least 3 experiments were less than 5% in all cases. Note the extra information which assists in interpreting the way the data in the plot was collected and displayed. This aspect of a good legend is also illustrated by a sentence in the legend for Part C, where we are told the BRCT mutants were grown to exponential phase and either mock-treated or UV irradiated at 50 joules per meter squared. The resulting protein extracts were blotted and probed with anti-Rad9 and anti-Rad53 antisera. Because Part C is a crucial experiment in this paper, this detail is important so that we can understand how it was carried out and how we can interpret it. As mentioned earlier, figures must always be referenced from the text, otherwise they're deemed to be superfluous to the discussion of the paper, and a journal will have them removed. Usually figures appear at the first convenient place after their initial mention. In a thesis or a written project where figures have their own leaves, this will usually be the subsequent page after the first mention. Let's look at how Soulier and Lowndes referred to their figures. In the middle of paragraph 2 on the first article page, we read, "A deficiency in arrest at the G2 checkpoint similar to that of the Rad9 delta control was observed, Figure 2b. See how the conclusion drawn from the observations displayed on the figure are stated, followed by the direct reference to where to check that point on the figure. These reference tags are written in brackets. We then hear, Western blot analysis of the mutant Rad9 proteins demonstrated levels of expression equivalent to wild-type Rad9, except for Rad9 delta CT, which was consistently expressed at 2 to 3-fold lower levels, Figure 2c. Rad9 is normally hyperphosphorylated after DNA damage, and this coincides with checkpoint arrest. In contrast to wild-type cells, none of the Rad9 BRCT mutants displayed any hyperphosphorylation after DNA damage, Figure 2c, or MMS, data not shown. And later, at the end of the paragraph, Western blot analysis of the mutants demonstrated that no Rad9-dependent phosphorylation of Rad53 occurred in the BRCT Rad9 mutants after DNA damage, Figure 2c. Notice how the figures referenced 3 separate times to substantiate different points. Notice also how the reference comes directly after the specific point which is being used to illustrate. In this screencast we've looked at how figures work as the foundation of good science. They provide the factual proof to substantiate findings and conclusions in the body of any scientific writing. Because they must be linked into the text, their labeling and legends must be clear and concise. Standard ways of doing this ensure all scientists can follow the results being presented. The test of a good figure is whether it is self-standing and can readily be understood by any knowledgeable reader.